of life begins with a single cell. But later it is no longer alone. It divides repeatedly to form a mass of many cells together. And in this video, we are going to learn about this beautiful process that helps us grow to what we are from a single cell. Yes, we will be talking about how the embryo develops. Do we have a scientific name for this process of embryo development? Absolutely, it's called the process of embryogenesis. In simplest words, embryogenesis is the process that follows fertilization. Where exactly does the fertilization take place in humans? Yes, it takes place in the fallopian tubes of the female reproductive system. However, before reaching the uterus, the embryo undergoes a series of divisions that help in its growth and development. Let us focus on one of the fallopian tubes where the egg cell has got fertilized. Now this fertilized egg cell will divide to form 2 cells followed by 4 and so on. This results in the formation of a ball-like structure. This ball contains many cells inside it. Do you know what this ball of cells is referred to as? When the cell number in it is 16, then it is referred to as the morula. This further divides to form a structure called the blastula. So will it have double the number of cells in it? Blastula is just a stage where the cells arrange themselves such that they form a hollow mass structure. In fact, the cells align themselves to the ends and leave a cavity within, which gets filled with the fluid. Now the next stage of development will be the blastocyst. It is the stage where the embryo will have cells that have got differentiated and will even differentiate further. Now what exactly is this process of differentiation? Let me explain. Have you ever thought that if we all started our lives with a single cell, then where do all these different types of cells in our body come from? Yes, I know it's from the single cell that was present at the beginning. But how is this possible? The answer to this is the differentiation process. As the name suggests, it is the process in which one cell gets differentiated from the other cells in its vicinity. As a result, we get a whole new variety of cells from these differentiated ones after successive divisions. So coming back to the blastocyst stage, it is now a mass containing differentiated cells. As it grows further, the body of the embryo begins to develop. But for its proper growth and development, don't you think the embryo will require nutrition? Of course, yes. And where will it derive the necessary nutrition from? You may say the mother's body. That's correct, yes. But how will that be possible when the blastocyst is still moving inside the reproductive tract? It is not connected to the point from where it can derive nutrition. Hence, the blastocyst needs to be connected to that particular location. This is where the most important stage of implantation comes into the picture. Can we guess what could it be from the name itself? Yes, it is the step where the blastocyst now attaches itself inside the mother's body at one fixed position. It implants itself there. And where will this position be? Of course, inside the uterus. That is the place where the baby grows, right? So the embryo now attaches itself in the uterus. Few of the main reasons for attachment is deriving nutrition, giving away wastes, and exchanging gases. This is served through this barrier that we can see here. This barrier is an organ called the placenta. So can we say that the placenta connects the baby with its mother? Absolutely. Now the embryo is all set to grow into a baby. The stage where the organs of the baby's body starts developing such that they can be identified is called as the fetal stage. And the embryo can now be called a fetus. The fetus grows in the uterus after taking the required amount of time, and when the growth is completed, the child is all set to enter this beautiful world. This is how an embryogenesis takes place in most of the animals. But wait a second. We have seen that a hen lays eggs and the chicks emerge out from these eggs. It means they do not grow in the body of their mother. Yes, development of the embryo takes place in 2 different ways. The parameter to categorize these types is the location of the embryo development. So how do we categorize the two types? It's simple. The first type is as we have seen in the case of humans. The fetus grows inside the mother's body. It results in the mother giving rise to the young one directly.
Hey everyone, I am Lapipon from BSET to A Science and I'm going to discuss the 4 stages of embryonic development. The 4 stages of embryonic development are: first is germinal stage, second is gastrulation, third is neurulation, and fourth is development of organs and organ system. The germinal stage of development is the first and the shortest of the stages of the human lifespan. It lasts usually 8 to 9 days, beginning with fertilization and ending with implantation in the endometrium of the uterus. After, with the developing organism is called an embryo. During this time, the organism begins cell division and growth. After the fourth doubling, differentiation of the cells begin to occur as well. Germinal stage has also 5 stages. First is fertilization, second is cleavage, third is blastulation, fourth is implantation, and fifth is embryonic disc. Fertilization happens when a sperm cell successfully met an egg cell in the fallopian tube. Once fertilization takes place, this newly fertilized cell is called a zygote. From here, the zygote will move down the fallopian tube and into the uterus. The zygote will burrows into the uterus lining. During cleavage, the cells divide without an increases in mass. That is, one larger single cell zygote divides into smaller cells. Each cell within the blastula is called a blastomer. Cleavage can take place into two ways: the holoblastic or the total cleavage, or meroblastic, the partial cleavage. During blastulation, cells continue to divide and begin to differentiate. The outer cell of the morula are polarized. That is, one side of the cell differs from the other side. The outer polar cells rise to trophoblast and the inner apolar cells become the inner cell mass. Implantation is a process which developing embryo moving as a blastocyst through a uterus make contact with the uterine wall and remains attached to it until birth. The lining of the uterus or endometrium prepares for the developing blastocyst to attach it to it via many internal changes. The formation of the bilaminar embryonic disc precedes gastrulation. As gastrulation progresses, the embryonic disc becomes trilaminar and the notochord is formed. Through the process of the neurulation, the notochord induces the formation of the central nervous system in the embryonic disc. The second stages of embryonic development is gastrulation. During gastrulation, cell movements result in a massive recognition of the embryo from a single spherical wall of cells, the blastula, into multi-layered organism. During gastrulation, many of the cells at or near the surface of the embryo move to a new, more interior location, the small circular region located at the cranial end of the primitive trach, where gastrulation occurs and is a controller of this process. The process establishes the three germ cell layer: the endoderm, the mesoderm, and the ectoderm.
Good day to everyone, I am Joseph Saison from BSED 2A Science. From the previous video that we have watched, Ms. Kwon discussed to you the first and the second stages of embryonic development. So I am now here to discuss to you, to present to you the third and the fourth stages of embryonic development. So the third stage is the differentiation. So in this stage, this is the third stage. So in this stage, it starts to make specialized structures and more function. So it is become more functionable. So the body now produce neurons, skin cells, muscles, and etc. in these stages. So then also in these stages is the organized into tissue, and the tissues organize into organs, and also the organs into organ system. So it is a process by which a tissue becomes organ and the organ become an organ system. So another stages, or the fourth stages of embryonic development, is the growth. So this is the last stage. This is the last stage of embryonic development. So in this stage includes the organism goes through the period of growth. So as what I've said, growth is more on the development or the progress happens in a body or in a cell. So in these stages also the new cells are formed and more extracellular matrix. So in these stages all complex development happens. So it is more on the development of it is a complete development rather.
Good day everyone, I am Mark Justin Salazar from BSED 2A Science, and I'm going to discuss about the embryonic development and their function. Before we discuss our topic about embryonic development, it means embryo. Then mostly embryonic development start with the fertilization of an egg cell, then just called an ovum, then by a sperm cell, then just called a spermatozoon. Once to fertilize, the ovum becomes a single diploid, or all known as a zygote. Starting in the second week after fertilization, the embryo start to develop distinct of all layer cells layers and form, although they will continue to develop and grow in the next stage, that's called of a fetus. Embryonic development is to develop these things of all layers. Then the function of embryonic period is all about the formation of important system of all the body. Embryonic development is to develop and start with the fertilization. Think of it as your baby's basic foundation and framework. So it means your babies from the mother is nag-work, then the fetal period on the other hand is more about growth and development, so your babies can survive in the outside world. In the last part, embryonic development will start with the fertilization in the egg cell, or just called an ovum. Then embryonic development and their function is to develop these things of all layers. And this is my topic about the embryonic development and their function. That's all po. Thank you.
environmental risk during embryonic development that I will mention is nutritional deficiencies in the mother. A maternal diet lacking certain nutrients may cause birth defects. The birth defects, called spina bifida, is caused by a lack of folate when the nervous system is first forming, which happens early in the embryonic stage. In this disorder, the neural tube does not close completely and may lead to paralysis below the affected region of the spinal cord. Thank you for watching this video.